Well, now I'm going to turn to the study of the response to high CO2, much of which happened here. The ventilatory response to PCO2 was long known to occur somewhere in the medulla. In the early 1950s, Isidore Lucen in Belgium had stimulated ventilation in dogs by acidifying their cerebrospinal fluid with acid. Hans Winterstein confirmed his results. Then Hans Luschke in Göttingen began searching for the site of the CO2 chemosensitivity. That was 1958. The most sensitive site he could find was in the lateral recesses of the fourth ventricle nearest the openings into the subarachnoid space. At UCSF, pulmonologist Robert Mitchell and Luschke read the Lucen and Luschke reports. In 1958, he injected mildly acidified mock spinal fluid into the lumbar subarachnoid space in two of his severely hypercapnic COPD patients with their permission. Within an hour, this stimulated an increase in ventilation enough to normalize their PCO2 from maybe 80 down to 40. In 1959, Mitchell joined my lab in the CVRI with Julius Comro. Comro told Mitchell immediately not to publish or continue his study in patients. I presume he was acting as the Human Research Committee all by himself. There weren't any at that time. Anyway, internist Mitchell then gave up being an internist and became a physiologist to hunt in animals for the brain surface ventilatory chemoreceptors responding to CSF acid. In 1959, I and the CVRI sponsored Hans Luschke to come from Göttingen to work at UCSF for a while with Mitchell. They worked together from September through November of 1960. Working through the dorsal exposure of the brainstem, they could not find a site. As a last-ditch effort before Luschke returned to Germany in late November of 1960, Mitchell slid a catheter in the subarachnoid space around the brainstem, like that, to the invisible ventral surface. He found a strong ventilatory response was generated by injecting slightly acid mock spinal fluid through that catheter on the inside or downside of the brain. Over the following months, Mitchell then developed a very difficult and bloody destructive surgical approach to the ventral surface of the brain through the palate of cats. In late 1961, he localized the superficial chemosensitive areas in the regions shown here by that black ellipse. It's around the anterior inferior cerebellar artery. There's one on each side, of course. Anatomist Alan Bassbaum here at UC had developed a new method to stain brain neural cells that had been very vigorously stimulated for at least an hour. The stimulated cells generated a substance called C-FOS, F-O-S, that Bassbaum could identify with a special stain. He helped Moto Sato, a Japanese anesthesiologist postdoc in my lab, to use this C-FOS staining method to locate chemosensitive cells in rats after 1 hour of severe stimulation by breathing 15% CO2. These black stain cells were found, as Mitchell's experiments had predicted, within a tenth of a millimeter of the ventral surface, thus being very exposed to the overlying CSF, extracellular fluid, and spinal fluid, and its pH. In this microphotograph, the C-phos cells were stained even when the animals were pre-treated with enough morphine to eliminate the ventilatory response to high PCO2. Morphine apparently does not block the C-phos generation in these chemoreceptors. Mitchell and I then wondered whether the human spinal fluid might in some way explain puzzles about high altitude acclimatization.